Hi everyone, I'm Chuck Wight, President of Salisbury University. As we prepare for the upcoming fall 2021 semester, the most important thing we can do to protect each other from the COVID-19 virus is to get vaccinated. This is especially important as the new variants of the virus are spreading rapidly among those who have not yet received the vaccine. Some people have been uncertain about getting vaccinated, especially with vaccines that employ the messenger RNA technology. So I thought it would be useful to explain how these work and why they are both safe and very effective. Our story starts with proteins, which are built from long chains of molecules called amino acids, and sometimes incorporate metal ions and other small molecules to help them function properly. This is a molecular model of the protein hemoglobin, which exists in our red blood cells to transport oxygen from our lungs to other parts of our bodies. Other types of proteins include structural proteins to help our cells keep their proper shapes, enzyme proteins that act as catalysts to help us digest food, regulatory proteins that help keep our body chemistry within certain limits, transmembrane proteins that pump nutrients and salts into and out of our cells, antibody proteins that recognize and bind to foreign substances or antigens to deactivate them, and many, many more. All of the different types of proteins are manufactured in the cells that make up our bodies. The recipe for these proteins is stored as DNA in the nucleus of each cell. The normal process is for this recipe code to be transcribed into messenger RNA inside the nucleus. The messenger RNA is then transported to the cytoplasm outside the nucleus, where it binds to ribosomes to ensure that proteins are manufactured with the correct sequence of amino acids. A virus is an infective agent consisting of nucleic acids, either DNA or RNA, covered by a coating of protein. We have all seen pictures of the SARS-CoV-2 virus, which is covered with spike or S proteins. The spikes are used by the virus to bind to ACE2 receptors on our cells, which allow the virus to enter the cells and multiply. Viral proteins, such as S proteins, are foreign to our bodies, so our immune system is able to recognize these proteins and manufacture antibodies that are specifically designed to bind to them, thereby blocking the attachment of SARS-CoV-2 to our cells and thereby preventing infection. But this process of recognition and manufacturing of specific antibodies can take up to a few weeks, and during this time, a virus can cause severe illness and sometimes death. That's where vaccines come in. A vaccine introduces foreign proteins into our bodies to stimulate the production of these specific antibodies when we're healthy. The classical approach uses dead or weakened virus particles. However, the newer messenger RNA vaccines use our body's normal function to produce just the spike proteins of SARS-CoV-2 without any of the other protein coat or nucleic acid material. Messenger RNA is short-lived, and it's destroyed by enzymes in our body as a normal part of the overall protein synthesis machine. However, once our immune system sees and recognizes the spike protein as a foreign protein, it produces antibodies that are prepared to attack the SARS-CoV-2 virus for a very long time. This process takes a while, which explains why we're not considered to be fully vaccinated until 2 weeks after our final dose. There are a couple of important takeaways from this simplified picture of how vaccines work. First, most people have a minor reaction to the vaccine, especially the second dose of the 2-dose vaccines. But this is actually a good thing, because it signals that our immune system has detected the presence of the spike protein and is starting to manufacture the antibodies that may be needed later to attack the real virus. I have a rare autoimmune disease, and I receive treatments that suppress my immune system. Last winter, I suspended my treatments for a few weeks in consultation with my doctor to get the Moderna vaccine. After my second dose, I ran a mild fever and felt pretty lousy for about 24 hours. But this made me happy because I knew it was a good indication that my immune system was responding appropriately to the vaccine. The second takeaway is that the messenger RNA in the vaccine never enters the nucleus of our cells and never interacts with our DNA. There is no alteration of our genetic material, and the only long-lasting effect of the vaccine is the memory of our immune system to produce the proper antibodies. COVID-19 has caused the death of more than 600,000 people in the US alone, and millions of people have died worldwide. The 3 vaccines approved by the FDA under Emergency Use Authorization are known to be safe and effective. The occurrence of adverse side effects is extremely rare, and the risks associated with those side effects are much smaller than the risks of serious illness from COVID-19 as a result of not being vaccinated. That's why we're encouraging everyone to get vaccinated against COVID-19 as soon as possible. At Salisbury University, we are looking forward to getting back to our normal, high-energy educational environment this fall. And we know that getting everyone vaccinated is the key to success for keeping us all healthy and safe. I'm grateful to Professor Mark Frana for assisting me in the preparation of this presentation. If you have any lingering questions about the safety and efficacy of COVID vaccines, please send them to stayinformed@salisbury.edu, and we'll try to get the answers for you. Thanks for watching.